So good morning, good afternoon, good evening, and welcome. I'm Susan Stern, and I have the privilege of being the CEO of Weizmann Canada. Thank you all for joining us and for your generous support. I'm grateful to have my colleagues and philanthropic partners from other countries joining, as well as members of the International Board. Thank you to the Weizmann Institute management and staff who are leading us through these challenging times and moving the research forward. Science will have the answer, and I'm so grateful to all of our scientists. I have a confession to make. I'm one of those people who gets all starstruck when they meet a celebrity, a famous person, a world expert. That's how I felt 15 years ago when I first met Professor Ruth Arnon, and how I feel each time I have the opportunity to spend time with her. Ruth is a brilliant scientist who's changed the lives of so many people around the world, and her impact on humanity will likely grow exponentially very soon. Yet she's so humble and gracious, she instantly makes you feel very comfortable. Ruth is an inspiration to all of us, and I'm thrilled that she'll be spending the next hour with us. Imagine the world without the flu virus. How amazing would that be? We've set aside time after her presentation for questions, and I'm grateful to have with us Dr. Arthur Slutsky, a member of our National Board, to moderate the Q&A. Dr. Slutsky is a professor of medicine, surgery, and biomedicine at the University of Toronto. He's a pulmonary and critical care physician with more than 500 peer-reviewed publications and over 75 book chapters. Until recently, and for the last 18 years, he was head of research at St. Michael's Hospital in Toronto. Dr. Slutsky is also chair of Clinical Trials Ontario and was recently named to the Order of Canada, one of the country's highest honors. We're very proud. His research interests include the primary role mechanical ventilation plays in the development of subsequent lung injury and multiple organ failure. So you can imagine that his expertise has been in very high demand during this pandemic. We're honored that he took the time and agreed to moderate this important discussion. I encourage you at any time to submit questions to Dr. Slutsky for Professor Ruth Arnon in the chat box at the bottom of your screen during and after the presentation. At this time, I'd like to introduce Professor Rowie Ozeri, who will say a few words about what's happening on campus and formally introduce Professor Ruth Arnon. Rowie is the Vice President of Resource Development, and as well as being a world-class physicist. Rowie conducts research in the field of ultra-cold atoms, a key component for quantum computing, which will make for a fascinating talk at another time. He's an excellent leader and also believes very strongly in the importance of science education. Rowie?
Thank you very much, Susan. So morning, afternoon, and evening to all of you. Thanks for joining in, and also thanks for being our friends and supporters and philanthropic partners throughout the years, and in particular during these challenging times. Israel is slowly getting, or not so slowly, getting out of the lockdown. People are not restricted to staying at home. More and more workplaces go back to being operational, and that includes the Weizmann Institute. If until about a week ago we were limited to less than 40% of our staff, currently everyone can go back to work on campus. We have what people here call purple badge, which means that we're sufficiently important to be allowed to hire everyone and let everyone go back to work. I have to admit, it's a good feeling in many ways. It's a good feeling because a couple of weeks ago, if you walked through the corridors, you could hear your steps and your steps only. Now you hear more and more people back doing science, doing research, talking about science. We still have to comply to the social distancing regulations, which means wearing masks, which means the number of people per room is limited. But Weizmann Institute is very quickly going back into its usual research mode to to as much as possible in these days. When I say that the Weizmann Institute is back to its research capacity, that does not mean that during these 7 or 8 weeks of lockdown, the Weizmann Institute was not working very, very strongly and fast towards research. But the research during these 7 or 8 weeks were very, very focused towards trying to come up with remedies to the COVID-19 situation. It was actually inspiring to see how probably already as early or mid-March, many groups across the campus, and I'm not talking only about groups that specialize in life sciences, groups from computer science, group from the math department, groups from chemistry, groups from physics, and of course our biology and biochemistry faculties immediately searched for a way to direct their expertise, to direct their talent, to direct their creativity towards advancing coronavirus research. I think that overall, I think the last time we counted was 2 weeks ago. We counted 66 different research projects at the Weizmann Institute trying to fight the coronavirus. I think that these projects can, grossly categorized into 4 different categories. One of them, which was fascinating, is our ability to perform coronavirus tests, either RNA tests that tell you whether the virus is in your blood, whether you're a carrier of the virus, or serological tests, which tells you whether your immune system has already developed antibodies against the virus. That means you have been infected, and now perhaps you're already immune naturally by having the disease. The Weizmann Institute actually turned some of the facilities in the Institute of Personalized Medicine, the INCPM, in order to help the national testing effort. Another domain of research is that of drug discovery. So several of the chemistry and biochemistry groups have accelerated their research and their expertise in finding small molecules that target viruses in order to find drugs that will alleviate the dangers of the coronavirus. Some of the groups are working towards vaccines. Some of the, and you'll hear a little bit about, or a lot about vaccine efforts from Professor Ruth Arnon. Some groups actually moved their research. These are groups that are based at the computer science department, use their computational and largely artificial intelligence capabilities to try and deal with the vast amounts of data that can be collected, either through tests or through questionnaires, in order to try and identify foci of outbreak of coronavirus disease in order to help decision makers make better informed decisions. And lastly, through the Davidson Institute, there was a strong effort, and still is a strong effort, to provide kids, students who are stuck at home, with means to use that time in order to get scientific education, and in particular, get more information about viral diseases, get more information about the COVID-19 virus, and that with the understanding that knowledge gives people, empowers people, and gives people the tools to deal with these uncertain times. So you have to, in order to, for us as management, in order to support COVID-19 research on campus, we had to come up with all the resources that are needed. That means lab space, that means core facilities that will provide the services that researchers need. That also needs funding, and that's why we started the Coronavirus Response Fund, which I know many of you have contributed, and I'd like to thank you from the bottom of my heart for supporting these research. Some of these researchers have already advanced, and I'm sure we'll be making a difference here, and you are allowing us to make that difference. So again, thanks. We now understand that this situation is not a short episode. We understand that we need to direct our efforts towards the longer term, both in terms of fighting vaccines for this virus, as well as fighting finding vaccines for other plagues that might be coming in the future. We understand that we need to deal with the mental health situation of people who have been locked down for such a long time. And that means that we're trying to, or we're now finding the ways to establish the means and the infrastructure that would enable research in these directions in the longer term. So having said that, I think that's a good introduction to introducing Professor Ruth Arnon, who's really one of the pillars of scientific research at the Weizmann Institute and in general. Professor Arnon earned both her graduate degrees from the Hebrew University prior to joining the Weizmann Institute in 1960. She's the incumbent of the Paul Ehrlich Chair in Immunochemistry, and Ruth really held almost every possible management and senior position at the Weizmann Institute, including department head, dean of the biology faculty, being vice president. Ruth made many significant contributions to the field of vaccine development and cancer research, study of parasitic diseases. She co-developed Copaxone, an MS drug, which made incredible impact around the world. And now, as you'll soon hear, Ruth is working on eliminating the flu virus from the face of Earth, which I'm sure all of us will be very happy with. Ruth Arnon also held many senior positions outside the Weizmann Institute, and I'm not going to list them all, but I'll just say that between 2010 and 2015, Ruth served as the president of the Israeli Academy of Sciences and Humanities, which is one of the highest ranking positions and honor you could find in the Israel academic landscape. Besides being an inspiring scientist, Professor Arnon is also a generous person, a philanthropist herself, and Ruth and her husband Uri made generous gifts to the Weizmann Institute, established the Arnon Science Education Campus. And for those of you who would visit the Weizmann Institute after this challenging situation is over, I invite you to come and see this amazing science education campus, which is on the southeast end of our campus. So with that, I would like to give the stage to you, Ruth, to tell us about your flu research.
First of all, I want to say something about vaccines in general. Before I talk about the vaccine that we try to develop for influenza. As you all know, there are many infectious diseases. And usually, when anybody contracts an infectious disease by a virus, caused by a virus, sometimes by bacteria, our immune system reacts against it, and it induces either immune cells or antibodies that are produced by them, and this eliminates the infection and give us protection for the future against the same infection. However, it was found, by the way, this is why there used to be childhood diseases, because usually children would have contracted the disease at childhood and then be protected for life against this particular disease. But it was found that it is not necessary to contract the disease in order to get protected. It is possible when you know the virus or the bacteria that is causing the disease to either kill them or attenuate them, and then use this product in order to, by injection, in order to protect us against the disease in the same way, because this killed or attenuated virus will induce a similar immune response that is induced by the disease, and thereby protect us against the disease either for the rest of our life or for a certain period, depends on the vaccine that is produced. This is in general. Usually, the portion of the virus that induces the immune response is the protein are the proteins that are on the surface of the virus, and they induce an immune response, and either the cell, the immune cells, or the antibodies recognize these surface components of the virus when they contract it and can kill it or eliminate it so that they protect us against the disease. Now, the influenza virus, I mean, in this way we have vaccines against many diseases, including measles, and using vaccines, several real killers have been completely eradicated, such as smallpox, polio, and measles is almost eradicated by vaccination against the virus. Influenza is a little different. It's different from most of the other viruses in the sense that it has two major surface proteins called hemagglutinin and neuraminidase. You don't have to remember the names. These are H and N. You know that the virus is defined as H1N1, H2N3, etc. The H and the N are two surface proteins of the virus, and they are involved in the activity of the virus. Now, a special property of the influenza virus, which differs from practically all other viruses, is that these two proteins change every year. Every year, they undergo changes in the structure and the sequence, amino acid sequence of these proteins. And because of these changes, the vaccine that has been produced against the virus of last year will not recognize the virus of this year, and therefore will not protect against it. And the vaccine that is produced against the virus of this year will not protect against the strain that will be the circulating strain next year. And this is why we have seasonal vaccines, because for every season we need a particular vaccine. Now, how are these vaccines prepared? Every year in April, about half a year before the season of influenza, the WHO, the World Health Organization, has monitoring stations in different places, mainly in the East, and they try to identify the circulating strains at that time, and they choose from them usually 3, sometimes even 4 different strains, and they they assume or predict that these will be the circulating strains in the winter to come, and they give instructions to all the vaccine producing companies that in this year vaccine, for this season, you include these 3 strains or 4 strains in the vaccine. So it takes now about half a year to produce the vaccine. Why? Because the influenza virus is grown or cultivated in fertilized eggs. So this is a whole procedure. You have to have enough fertilized eggs in order to infect them with the virus, and from them isolate the virus, attenuate or kill the virus, depends on the type of vaccine that is being prepared, and then you prepare enough of it in order to be sufficient for millions or tens of millions or hundreds of millions of people to be vaccinated just before the season. So you have to start working on it in April so that in October you have enough of the vaccine to be used as a seasonal vaccine for this particular season. Next year, the whole procedure starts again. Now, we have taken a different attitude, and for this I have to go back to my research that is basic research that is done in order to understand the basis of the chemical basis of antigenicity. Now we know that here that there are two proteins that are for all viruses. The component of the virus that are responsible for the infection, and therefore the vaccine is directed against them. Usually these are proteins. We were interested in the immunological properties of proteins. And the question that we asked ourselves, and I'm talking now about years back, is it really necessary to immunize or vaccinate with the entire molecule of a protein in order to get an immune response against the protein? Or is it possible to take just a part of the molecule? If we can identify a significant part of the molecule and use it either by taking it out or producing it from the virus itself, or by preparing it synthetically, and use this fragment of peptide in order to vaccinate or immunize and can we get protective immunity? So we started years back with, first of all, with a simple protein that we knew the structure of, and we, looking at the structure, we predicted that a particular fragment of the protein would induce an immune response that will recognize the entire protein. So we synthesized this fragment and used the synthetic fragment, and indeed, we got a very good immune response against the protein. The protein that we worked on was a material called enzyme. Enzyme is a material that has protein that has biological activity. So we could test whether the antibodies that we have produced have any effect on this biological activity of the protein. And what we saw is that using different peptides of the same protein, we could induce antibodies that either can neutralize the biological activity of the protein or other antibodies that would not neutralize. So we have a control. We can select particular parts of the protein that has biological activity, and we can induce particular antibodies that will inhibit this immunological activity. Okay, so this was a very interesting research project, but now we asked ourselves, is this limited to just a purified protein, or will the approach work also against a protein which is a part of a virus? So in the beginning, as a model system, we chose a virus that does not infect people or animals. It infects bacteria. It's called bacteriophage. This is a virus that infects bacteria or affects bacteria and kills the bacteria. So we used the same approach. You looked at the surface protein of this virus, of this bacteriophage, and we predicted that a particular fragment of this protein will induce antibodies that can then inactivate the protein or kill the virus, as a matter of fact. So indeed, we synthesized this fragment of the protein. Again, I emphasize, this is a protein that is a surface protein of a virus that kills bacteria. So we synthesized this peptide, we attached it to a particular carrier, immunized mice with it, or rabbits, isolated the antibodies that they produce, and we found that these antibodies could indeed inactivate or kill the bacteriophage. So the bacteriophage could not kill bacteria anymore. Okay, nobody is interested in a vaccine against a virus that infects bacteria. So we wanted to see whether the approach will work also against a virus that is of more interest to us. And we selected influenza. I'm talking now about many years ago, not because I had an idea of producing a commercial vaccine against influenza, because so much was known about influenza virus and its various proteins. So we thought that it was a good model for us as a virus to study. Now, influenza is different from most viruses in the sense, as I said, that the two proteins on its surface are changing every year, as I said before. So we had to look at these two proteins and to select which fragments of the protein we want to synthesize and to immunize with so that they will induce a protection against the virus. So our idea was not to focus not on the regions of the proteins of the virus that change every year, but on the contrary, to focus on regions of the virus that are conserved against among many strains of the virus. First of all, because I think that maybe regions are conserved because they are essential for the virus. This is why the virus doesn't change them. If they change, the virus dies or stops to be infective. And we looked at the virus and we identified, first of all, one such fragment that is a conserved region of the hemagglutinin molecule, of the H molecule of the virus. We synthesized this fragment. We attached it to a good carrier and immunized rabbits with it. We took the antibodies of the rabbits and we saw that the antibodies can actually inhibit the virus. So we immunized mice. Now mice are sensitive to influenza, to the same strains of influenza that are infective in men. They are sick with influenza, and if you give them a sufficient dose of the virus, they would die of the infection. The infection, the virus affects the lungs, and you can find the virus in the lungs and in the serum in the blood of the mice, and they get influenza. So we immunized mice with our construct, and we saw that we get 30% less infection in the mice that we immunized. We immunized the mice just with the construct that contained one small fragment of the coat protein of the surface protein of the virus, the H protein. Okay, so I said maybe we can improve that a little bit, and maybe we can add some other fragment. And we predicted two other regions of the proteins of the virus that will induce not antibodies, but that will induce cellular immunity in mice. And we had altogether 3 different regions of the virus. One of them induced antibodies, two of them induce cells, but one is a particular cell immunity, cytotoxic immunity. So we immunized with a mixture of these 3 peptides, and we got complete protection of mice against the infection. All the mice in the control group, when we immunized, we had a control group, and afterwards, we subjected all of them to little dose of the virus, of the infective virus. And what we found is that all the mice that were in the control group, all of them died from the infection, and 100% all the mice that were immunized with the combination of the 3 peptides survived. Those that were immunized only with the one, only part of them survived. Those that were immunized with combination of 2, and we checked all the different combination of 2, were partly protected. And those that were immunized with the 3 peptides were completely protected and none of them died. Okay, that's wonderful. So we have a very good vaccine for protecting mice against influenza. How do we go from that to people? So we used a model, an animal model, that is called humanized mice. What are humanized mice? These are mice that by 2 doses of irradiation, total body irradiation, you actually kill their entire immune system. They, if you leave them, they'll probably die because their immune system is dead. Then you transplant them with human white blood cells. So now they continue to be mice, but their immune system is a human immune system because all their white blood cells are human white blood cells. So this is a good model for studying how these humanized mice will react against or the immune response that they will produce, because it will be similar to or we think that it will be similar to the immune response that human will put up. So in this case, we had to choose other regions that will activate the human immune system, and we had 4 different peptides. And again, here we prepared them and we studied each one of them individually. In short, combination of the 4 peptides that were presented in a certain form, when we immunized with them, we could protect the humanized mice completely against infection. They were completely protection. Not only that, when we looked in their blood, we saw that they had antibodies. Both in their blood and in the lungs, they had antibodies. They had human antibodies that are specific against influenza. So we knew that indeed we could induce in these mice a human immune response against influenza. So and as I said, they were also completely protected against influenza. Not only that, I said that they had human antibodies. What we saw is that these human antibodies reacted against many strains of influenza. They did not react just against one strain, but against many strains of influenza. So, and when we tried to infect them with different strains of influenza, they were protected against any strains that we have started. So actually, this was as far as we could go in our laboratory. We published a patent that a construct like this could serve as a vaccine against influenza. And I thought that with this, I finished my research again for an influenza vaccine. But at that stage, YEDA always publishes the patents that are available at the Institute. And a group of 3 young entrepreneurs found interest in this project, and they have decided to establish a company that would produce a vaccine, a human vaccine against influenza that will protect not against one strain, but against many strains of influenza. And indeed, they took over. And first of all, they decided that they will not, that the 4 regions that we had are not enough. So they added, in consultation with me, they selected 5 additional regions of the virus, of the viral proteins. So they had 9 different proteins. Now, of course, they couldn't use a mixture of 90. We used in our studies a mixture of four peptides, but 9 for vaccinating people, a mixture of 9 different is a recipe for failure, and nobody could get anything approved by the FDA or any other agency for a mixture. So they produced actually it's a synthetic protein that is produced by genetic engineering and contain all these 9 different regions, each of them produced in 3 copies so that it will definitely induce a good immune response. And they immunized mice. They used, first of all, they checked it in mice. They used transgenic mice that express the immune components of the human immune components. And what they found is that they can get practically 100% protection. So mice that were not immunized, or mice that were immunized with any placebo control, if they used groups of 20 mice, then 18 or 19 would die after being infected, they would die of infection, whereas the mice that were immunized with their construct, 100% were protection, 100%. Not even one was infected. So now was the stage in order to start looking whether it will be effective also in people. So first you do, there is a sequence. First you do a phase 1 trial. First you do toxicity. You show that in animals it is completely safe. Then you do phase 1 study in which you immunize with a certain number of volunteers, and you show that the vaccine is completely safe and does not induce any mal-effects or adverse effects. Only after this stage is over, you can do what is called the phase 2 study, taking a relatively small number of volunteers and vaccinating them, and look at their immune response. And the company did here in Israel 6 different phase 2 trials like this. Each one in the beginning it was several dozen individuals, afterwards in several hundred individuals, so that altogether I think they vaccinated more than 600 individuals and tested them compared to the placebo injected, and they saw that they get very good immune response, both antibodies and cellular immune response. Cellular immune response meaning cells that can actually kill the virus. However, in these phase 2 trials, they did not test whether these particular individuals were protected against infection, because challenge what, by the way, now in the case of corona infection, they say that they may allow trials with challenge. Challenge means that you expose the vaccinated individual to the virus and see whether he or she is infected or not. In the case of influenza, this was not allowed. So the only thing that could be tested in this phase 2 trial is whether the vaccinated individuals produced antibodies that recognize a virus, and also immune cells that could kill the virus. And this was shown. And at that stage, they were allowed to do a phase 3 clinical trial. Now, this phase 3 involves 1,200 individuals, 4,000 individuals. Half of them are vaccinated with the vaccine, and half of them are vaccinated with placebo. 4,000 were vaccinated last year, and 8,000 this year. Now, it was required that this will be done in 2 years, so that because if you do it only in 1 year, and even if you show very good protection, it's only against the strain of this year. But if it will be effective in strains of 2 consecutive years that are not related to each other, then you can claim universality. So now we are at the final stage of this phase 3 trial. And at the end of this month, this will be the end of the influenza season in Europe. The trial, by the way, is done in 7 different countries in Europe, in Eastern Europe. And at the end of May, the trial will be closed. All the samples from last year were already tested, but from this year will be tested and will determine who has had influenza. By the way, during the trial, each individual participated in the trial got a call from a nurse every week asking, did you have fever? Did you have headache? Did you have did you have a cough? Did you have this? And if they had any symptom, then the nurse would come and would take a sample from the nose or from the throat. And these samples were kept until now, until the end of the season, and then checked, and and then they were subjected to determination whether they had influenza or not. And at the end of the trial, at the end of this month, we'll have all the samples available. I hope that it will take not more than a month or a little bit more to test all the samples. And then we will compare then the results will be open, and we'll compare the placebo versus the vaccinated and see whether it is effective or not. If it is effective, then it has a good chance of protecting against many, many strains of the virus, and it will be considered as a universal flu vaccine. And eventually, it will not eliminate the influenza virus, because the reservoir of the influenza virus is in birds, and birds can host the virus and are not infected. They are not sick. So the virus will continue to exist, but people will be vaccinated and protected.
Wow.
We hope for the best.
Amazing. I know there are a lot of questions, so I'm going to turn it over to Dr. Arthur Slutsky to moderate the question period. Amazing, amazing.
Thanks very much, Susan. Thanks very much, Ruth, for really, can you hear me?
Yes, Kev, I can hear you.
That was a really fascinating, you covered a lot of material in a way that I think is very understandable to the lay public. So kudos to you, and immunology is quite difficult. So there's a number of questions that are coming up. I had one to start, and I think you've sort of answered it, is why does it take so long to develop vaccines? People are thinking about that now, of course, largely because of coronavirus. And so why does it take so long? And I think you explained some of it. And you think that can be shortened now in terms of developing a coronavirus vaccine?
I think that the coronavirus vaccine will not have the intention will be just to have against these particular strain of the corona. So in order to develop a vaccine, you have either to kill the virus or to attenuate it in a way that it can still lead to protection without causing infection, or to focus on a particular protein of the virus. In the case of the corona, there is the spike protein that I'm sure that everybody has seen all the pictures of the virus with the spike protein that is the single surface protein protruding surface protein of the virus. And I think that all the efforts are now in this direction, either on the spike protein or on attenuating or killing the virus in one way so that it can lead to protection without leading to infection. Now, as I explained before, usually for a vaccine, you need 3 stages. Phase 1 for safety, you have to show that it is safe in people. Phase 2, that in a small number of individuals, showing just showing efficacy, but in a small number, and only then going to a large number and wait for them to be infected and compare placebo control with the vaccinated. I guess that in the case of corona, because the issue is so urgent, maybe they are talking now about allowing challenge trials. Challenge, as I said, means to vaccinate. If you have something that you think that it is active, you vaccinate, and 2 weeks later, you come, or I don't know, 2 weeks later, 3 weeks later, I don't know, as long as it takes for the immune response to develop, and then you give a challenge infection you subject the volunteers to the virus and see whether they are protected against infection or not. And this may shorten the time for developing the vaccine. But still, it will not take weeks, it will take months, if not more. I think that it will take at least a year and a half until we have a vaccine.
Yeah, I think that's, going from what it's taken you 10 years or so. Is that right? Have I got the numbers right?
Depends where you start to count.
Yeah, I think one of the more important messages actually from your talk was how you started to do this basic research without even thinking about how this might be applied to humans.
Not at all.
And now, if this works, this is incredible. So I think that's very important for the people to understand that basic science research, that's curiosity driven, is so important.
Mm-hmm. I want to emphasize this point because I really believe that in universities and in research institutes like the Weizmann Institute, the research that is being done should be basic research, and I define basic research by research that the purpose of which is to increase the amount of knowledge. Just for what you said, curiosity-based, to know more. But sometimes you step on something that may have some application. It would be stupid to say, well, this can be applied or applicable, so I don't touch it. On the contrary, I think that in case like this, you should push it further as much as you can. And by the way, the same thing happens in our research in the development of Copaxone. We did not intend to develop a drug. We wanted to study the mechanism of multiple sclerosis, and we worked on an animal model, and we synthesized the polymer that we thought that will induce the disease, but it did not induce the disease. It rather inhibited the disease. And therefore, step by step, we got to the development of a drug. But we started with a completely basic research that had no intention for application whatsoever.
So one of the questions that's come in, Ruth, is that this virus, the coronavirus, let's talk about that for a moment, has been around for a long time. And it's been known for a long time, at least probably 50 plus years. Why hasn't a vaccine been developed already to the coronavirus? And does that mean that it'll be more difficult than we think to develop one for SARS-CoV-2? Coronavirus,
actually it's a family of viruses that we've known for many years. Usually they cause what we call common cold, which is a short lasting disease, and there are quite a few strains of it, and they are not virulent or violent. So there was no interest in develop, and it's not the same one. They may differ from year to year. And it's a family of viruses, and nobody was particularly interested. But there were already two violent or virulent strains. One was the SARS and the other was the MERS. They were several years apart. And now is a third one that is very violent, more violent than both the SARS and the MERS. So now both in the cases of the SARS and the MERS, there were attempts to produce a vaccine. I think that there is a vaccine. I don't know how effective it is against the SARS and the MERS, and nobody knows if it is effective against the coronaviruses because nobody has tried it against the corona for vaccination. I'm sure that now there are some trials in these directions also because these vaccines exist. I don't know how effective they are, and people are working now for producing a more specific one against the COVID-19.
One of the questions that's come in, Ruth, deals with, I'll call it the basic science of immunology. And you mentioned the cellular immunity and innate immunity. Could you explain that concept to the lay public? What about the immune system? I know you only have a couple minutes to do this, but I think it's something that's an important thing to teach in terms of the immune system.
Okay, first of all, the innate immune system, and this is each one of us has a built-in immunity. We have there are cells of the immune cell of the immune system. There are two different kinds of cells that are called B cells and T cells, and both of them, some of them produce antibodies, some of them act by themselves on the disease-causing agent, and we have these cells that are not specific for a particular agent in our immune system. This is part of our immune system, a natural component in our body. Now, when you immunize with a particular agent that is done intentionally, then you induce specific cells, and some of these cells, one type of these cells, produce antibodies, and both the cells and the antibodies are directed specifically against the material, or it could be a disease-causing agent that, and the immune response is against this particular agent. So the innate is a native one. The acquired is an immune system that we induce by immunizing with something, and the result of the immunization or vaccination is that we have both cells that can recognize the invading organ and eliminate it, and antibodies that can react against it and neutralize it.
Super.
Does this answer the question?
Yes, thank you. You must be on Spielkes waiting to hear the results, because you don't know anything about the results as yet, right? You're blinded until the end.
In Spielkes is nothing. Half my fingers are broken from holding them.
I can see. So let's say the study is positive. Let's say the phase 3 trial is markedly positive. How long will it take to get this vaccine out to millions, potentially billions of people?
Well, the company in Israel has a facility for producing, I don't remember if it's 20 million or something doses a year. I don't remember the exact number. Anyway, it's in tens of millions. Now, in order to produce the vaccine for the entire world population, of course, a larger facility or several larger facilities will have to be made. But I want to emphasize the advantages of such a vaccine compared to other. First of all, you will not need, you can be vaccinated all year round. You will not have to be vaccinated just before the season, because whenever you want to be vaccinated, so there will not be such pressure on having the vaccines prepared in October, because you could be vaccinated in April. Once you are vaccinated, the vaccine, we know from mice that the protection lasts for at least 7 months. And 7 months in mice is equivalent to several years in people. So I guess that once you are vaccinated in the first place, then you will need a booster injection maybe once every 5 years, like tetanus. Mm-hmm. And so it will be like all the other pediatric vaccines that exist, and from time to time you need a booster injection in order, or the measles vaccine, it will be the same thing.
There's a question that came in largely related again to COVID-19, and that's the concept of herd immunity. Could you tell us a little bit about that?
There is a theory. Well, we know for about, in the case of many diseases, we know there is a phenomenon that is called herd immunity. We know that if 70 or 80% of the population are vaccinated against a particular disease, then the 20% or 30% that were not vaccinated are actually also protected, because there will be less of this disease, of this infection altogether. So everybody is protected, although only 70 or 80% were vaccinated. And this is called by the nickname herd immunity. Now, I don't know, and I don't have any, we don't have any fact of a disease or plague like this, that with of the corona, that when 80% were vaccinated, all the population were. This is only an assumption that nobody has tested.
Super. We're coming to an end. Ruth, turn this back to Susan. Let me personally thank you for a very fascinating hour. Next time we have you, we need you for a couple of hours, because there's so many questions that have come in, but we do have to finish on time. So thank you very much personally.
Then I can promise you one thing: if the trial is and we have a vaccine, I'll go and shout it all over.
Good, and I'll be the first one to take it. So send some to Canada.
And it'll be a great birthday present for you, Ruth. You know, I'm very rarely speechless. I think my husband is around here somewhere. Yes to that. But hearing this presentation really, I'm just so excited. And I have to read one of the comments that was sent to me privately from Steve Meyer. Sorry, Steve, you'll forgive me. When he started at Weizmann, he was a member of the senior leadership team at the American Committee. And he writes, when I started at Weizmann in 1995, Ruth Arnon visited our New York office just after getting Copaxone approved by the FDA. We had a champagne reception to celebrate. Today, I am feeling the same sense of exhilaration and pride. So thank you, Steve, for that.
Tell him that I'll come to New York to have champagne with him if the universal flu vaccine will be approved by the FDA.
Amen. And we're gonna hold you to that all around the world. So first of all, thank you all, everyone here, for making the time to be with us for this fascinating presentation. Thank you so much, Ruth. Thank you. We're so grateful.
Yeah, I'm very pleased. I hope that that what I presented was clear to the people who listen.
Metsuyan. It was perfect. And thank you for accommodating my wish for no slides, because I really enjoyed hearing you present the science. Art, thank you for masterfully moderating the Q&A. Listening to you, Ruth, fills us with hope. And speaking of great information, I encourage all of you to stay connected with your Weizmann family through the website of your local country. As you can imagine, research is moving forward at an unprecedented speed, and information is updated on websites regularly and often. Roe and the rest of the management team, thank you for your leadership. I'm sure when you signed on for this, you didn't anticipate what your first year would look like as a vice president. I also want to thank my team and the team at the Weizmann Institute, headed by Kelly Avidan, for their efforts in putting this and other events like this together. We're sending warm wishes for good health to all of you and your loved ones, and I look forward to being together soon on the campus of the Weizmann Institute. Thank you, and be well.